Ladies and gentlemen, it's my great pleasure to welcome everybody. This is the second time we are having this symposium. We had one last year that was very successful, and we followed on that, and we are very grateful to the funding agencies that have actually given us the funds to be able to do this. And there's three important partners that have actually contributed towards this. There is KEMRI-Wellcome Trust in Kilifi, there is CDC, and the USAMRU-K. I really am grateful to KEMRI for just really the leadership they show towards science in this country.
I am delighted to be part of the second Medical and Veterinary Virus Research Symposium. I see that this brings together both the human and veterinary scientists. This is really key as we emphasize on the One Health concept in the prevention of diseases. We know that most of the viruses are zoonotic in nature, and therefore bringing the scientists in these two fields will bring the much needed synergy in addressing prevention and control of these diseases. The MVVR initiative aims at mentoring young virologists, which is important to ensure the much needed capacity in terms of expertise is created. I have no doubt that this will be a very productive symposium, and therefore wish you very fruitful deliberation in the next 2 days. I therefore have this singular pleasure to declare the second Medical and Veterinary Virus Research Symposium officially opened. Thank you.
For the benefit of those of us who are not familiar with arboviruses, these are a set of, or a group of viruses that are essentially transmitted by vectors. The key vectors are mosquitoes and ticks, and sometimes we have some that are transmitted by sandflies. They are transmitted in a biological cycle that involves the vectors and the animals, and often human beings get exposed to these viruses as incidental hosts, and that then ends up causing disease in humans or outbreaks. And some of these viruses, like Rift Valley fever, also cause disease in animals, and we end up with losses in our livestock.
Today I'm just going to talk about the outbreak response and investigation activities that we've carried out since October 2011 to date. But we have various mandates, including research on arboviruses and viral hemorrhagic fevers. We also do surveillance, both in human populations and entomological vectors. And we also provide diagnostic support, both locally and regionally for various viral hemorrhagic fevers. And we also undertake training. Different countries have actually requested that we train their personnel in different aspects of diagnostics. When it comes to our capabilities, we do cell culture. We have both mammalian cell lines and also insect-derived cell lines. We do molecular diagnostics, both the conventional and the real-time PCR, and also we do serology.
The Rift Valley fever virus is a zoonotic disease, as we heard. It occurs in Kenya, and we have two different kind of cycles. The one is an endemic one. There mainly Aedes mosquitoes are responsible for maintaining the virus in the environment. So they somehow lay infected eggs in the soil, and these eggs can persist over a long time until rain is occurring, and kind of a flooding of these mosquito breeding habitats takes place. Then mosquito can hatch and start the cycle again.
We actually undertook a pilot study to determine the prevalence of hepatitis virus as well as genetically characterized HAV in children. And this study actually came about because we did participate in a cholera outbreak in Cameroon in 2010. And after the outbreak, some pediatricians were actually interested in finding out what was going on in some of their patients. They actually recorded some cases of jaundice and cholera, but they were not sure what was causing this infection. So they contacted us, and we decided to look at a couple of children who presented with diarrhea in some of their health facilities.
My story with oncogenic viruses goes back to the days I was working with HIV, and we had Kaposi's sarcoma in the late 80s. We didn't know whether it was a tumor or a hyperplasia. And finally, we confirmed it was a tumor. And it's the first virus for which we have a definitive viral cause.
Endemic Baxter lymphoma is childhood cancer, which is common in regions where we have high malaria transmission and also the presence of Epstein-Barr virus. It's known that the two infectious agents, PF or Plasmodium falciparum and Epstein-Barr virus, they actually interact together, probably leading to presentation of endemic Baxter lymphoma. In children with Baxter lymphoma, there's actually minimal, or if not, there are no responses or interferon gamma responses to endemic Baxter lymphoma, to EBNA1 peptides, which is actually crucial for Epstein-Barr virus.
Cancer of the cervix is the second most common cancer among women worldwide, with an estimated 500,000 new cases annually and around 274,000 deaths annually. That is according to WHO. And current estimates indicate that every year, around 80,000 women in Africa are diagnosed with the cervical cancer, and among those, around 53,000 die from the disease. And in Kenya, the estimates indicate that every year, 2,454 women are diagnosed with cervical cancer, and 1,676 die from the disease.
My work is based on contact patterns, but we want to know the ones that are relevant for the transmission of respiratory infections. And that is part of the work that I'm going to present. And we are going to put this data into a mathematical model and see what would be the effect of differences in the assumptions in the contact data. There are a number of determinants which are important in the spread. So one of it is the duration of infection. The other one is the number of contacts per unit time that a person will have who is infected. And then another one is the probability of infection for each contact with a person. But not all contacts actually are with infected or susceptible people.
RSV is an important viral infection in childhood and among the elderly, where the burden estimates are quite high. To date, no safe and effective vaccine has been licensed, although people do get immune to natural infection, and that natural immunity is strongly correlated with neutralizing antibody. The potential of future RSV vaccine interrupting transmission is quite limited because population-level neutralizing antibodies do not persist for a long time. So there is opportunity for recurrent reintroduction of the virus into the population.
Rabies is an acute viral encephalomyelitis caused by rabies virus. Transmission is mainly through saliva of an infected animal. An incubation period varies, especially in humans, ranging from 5 days to 7 years. And worldwide, more than 10 million people are infected, and more than 70,000 of these people ultimately succumb to rabies infection. On January 28th this year, we received information that a dog in Kisumu had bitten 8 people. And this information was actually relayed to the district veterinary officer. So upon investigation, the district veterinary officer euthanized the dog, decapitated the head, and sent it to the Central Veterinary Investigation Lab in Kabete. And on 2nd of February, the results came positive that the dog actually had died of rabies. So this actually is what triggered the outbreak investigation.
I just wanted to reorient ourselves to the fact that influenza A host diversity is quite broad, with mainly avian and also mammalian species. I borrowed this figure from Google to illustrate to us the interspecies transmission that occurs and also the host diversity. So the bold lines that you see between poultry or avian species and the humans represents the one-way transmission that has been established for influenza viruses, particularly H7 and H5. Now, if you look at between the humans and the pigs, you have the two-way transmission, which normally occurs. We have influenza viruses that are cross-transmitted from humans to pigs and also from pigs to humans. And this was clearly illustrated, most of us would remember, during the pandemic.
Bioinformatics as a field seeks to analyze large sets of biological data, to solve biological questions, and formulate underlying hypotheses that occur in a given biological sample that you are looking at. So why bioinformatics at this stage? It came into being because of this exponential growth of genomic data. As you realize, from the very early beginnings in the 1965, we had only few sequences or genomic sequences. But as time went by, we moved from those looking at single genes and went into high throughput analysis. So because of the bulk of biological data that was lying in the databases without any information having been assigned to them, then they needed some computational approach to really annotate or give biological meaning to the data that had been generated by then.
Sexually transmitted infections are actually the agents themselves. So when we are going to talk of the bigger picture, rather than talking of STDs, which are sexually transmitted diseases, one should begin to talk about STIs. And then now within those STIs, we can now talk about what? STDs. Sexually transmitted diseases as those diseases actually that after that conduit, then they present themselves at the genital organs. A good example is hepatitis B and HIV as well. They use the genital organs as a conduit, but they don't have any genital manifestations whatsoever.
The HIV virus is actually very highly replicating. It doesn't have an exonuclease enzyme for checking for any mutation that could have formed. So the virus replicates very fast. And there's also high recombination, so the transmission also of viruses from one region to another. This has led to high diversity of the virus. This I'm trying to show you the migration of people, and this is how people actually get HIV infections. And you see the HIV subtypes are distributed worldwide, with other countries having more predominant, like in US or Europe, you have HIV subtype B being more predominant, but in Africa, all HIV subtypes are actually circulating, and most of this is in West Africa. And as you can see, people migrating from one region to another. If you are infected and you infect another person, you also acquire the subtypes from that region as well.
What I'm going to discuss today is on a disease called African swine fever. And the question you need to ask on this is, why do we need to research on African swine fever? African swine fever causes heavy losses to pig farmers, and given the background I've given you, pork production globally is very critical if you want to feed the world population. With this disease, almost all pigs that are affected or infected die very fast within a very short time. And thus, from the death of pigs, this becomes a constant food security among smallholder farmers who keep these pigs.
In Africa, 70% of chicken production is under the indigenous, or what is known as the free-range system. When you go to the Western countries, you find that the intensive, commercial or semi-intensive is more common. And this is also reflected with the types of diseases that we find important. Like in Africa, Newcastle is economically important according to the OIE. Whereas when you go to Asia and Europe, you find that it's more avian influenza and other diseases that are more common in containment. This virus belongs to the family Paramyxoviridae in the Abulavirus family. It is a negative single-strand RNA that encodes for 6 genes, which go on to produce 6 proteins that are very important for the virus survival. So vaccination for the Newcastle disease is effective. The only problem in Africa, it is very expensive because it's under cold chain.
Rift Valley fever is one of the listed diseases. Apart from it being zoonotic, it also hampers trade, and it also causes production losses when there are abortions, and also when it causes death. Previous outbreaks of Rift Valley fever that we've had, we've seen that there's a lot of loss of institutional memory, or when you have an outbreak, people will run, work on Rift Valley fever, up to until 1997, and the last outbreak, 2006 and 2007, is when people have come up at least to document what is happening and even share data between the medics and even the vets. And actually, those are some of the things that brought up the zoonotic unit between the departments, apart from now avian influenza. Rift Valley fever has played a very important role in that.
Makere University Water Read Project initiated surveillance activities for influenza in 2008. This was following the 2005 outbreaks of the H5, and specifically wanted to do long-term surveillance, create capacity for long-term surveillance so that we are able to rapidly detect emerging pandemic strains and generate sufficient data to inform policy, and also to be able to respond to outbreaks. And indeed, because of these efforts, we're able to respond a little bit more quickly than we would have done to the pandemic 2009 outbreak.
Let me come to pandemic H1N1. This was initially detected in Mexico in April 2009. We realized that it is a distinct virus as a result of reassortment, and there was sustained human-to-human transmission, and that's why in June, the WHO declared it as the first pandemic in the 21st century. And Kenya reported the first case the end of June, beginning of July 2009. And since then, we have noticed that pandemic H1N1 has actually replaced seasonal H1N1 and has become quite a dominant virus.
The pathogens and the testing that we currently, we primarily focus on are all the Ebola strains, Marburg, Rift Valley fever, and CCHF. We have the ability to do more, but these are the ones that we focus on because they're the most likely that we're going to see here in Uganda. We do antigen detection ELISA, IgM, IgG, and also RT-PCR. So we have all 4 of these tests for all of these pathogens in the lab. As I mentioned before, we have an upgraded BSL-2, BSL-3 laboratory, so we have many different rooms, but the whole laboratory is dedicated to just hemorrhagic fevers. We also increased our biosecurity, biocontainment. We're developing an in-house LIMS system for the laboratory for sample management and data management. And also since 2010, we've processed over about 1,000 clinical samples. It's probably closer to about 1,500 now just because of the last outbreaks that we've had. But this represents over 15 districts in Uganda and also 4 East and Central African countries that we also serve as a regional referral laboratory.
Sicilian has been detected again in the Mediterranean region, in those countries including Egypt, Pakistan, Saudi Arabia, Iran, Cyprus. It has extended to Greece, with peak within the summer in endemic regions. Serosurveys have shown that Naples has been detected in Omo Valley, the Afars and Isas of Ethiopia in the 1970s, specifically 72 and 74, with no Sicilian documented.
Dengue is caused by arbovirus, and it's transmitted by mosquitoes, mainly of the Aedes aegypti. It causes acute febrile illness. In Kenya, there's very little information about dengue, and also in Africa. The first outbreak having been confirmed in 1982 in Malindi and in Kilifi, and it was caused by dengue serotype 2. Surveillance is also limited, and there's also lack of diagnostic capabilities.
Bioinformatic tools are coming in handy to assign or annotate the functions that is encoded in those reads that come out of the sequencing platforms. So when we look at metagenomics, actually we are trying to fit puzzles, because when you look at an entire environmental sample, you get a mixture of virus sequences of virus species or populations that are encoded in there. So you go in and try to untangle the genomes that are present and then try to fit the jigsaw so that you identify which virus populations are present or predominant in given environmental sample.
Mass-TAG PCR consists of it's multiplexed of many viruses, and the primers are labeled with mass codes, mass weights. So you see, if there is Rift Valley fever in a mix, then primers that are specific for universal for all fleboviruses, if I label them with tags that have specific numbers, let's call them here A and B. If Rift Valley fever is in the sample, it will amplify, and the tags will be on the end.
The aim of the conference from the beginning was to involve viral research in its broadest guise. The One Health concept, I think, is something that we need to take forward into the future. So what we've seen already is the regionalisation of this meeting. So it's opened up since last year, and I think the quality of the reporting, I thought last year was very good, but this year I think has actually improved considerably in terms of the presentations and the diversity of presentations. And the regional nature of it, the talks we've had from Uganda, from Tanzania, as far as Cameroon, it shows how much interest there is in this symposium, in this work, this sort of forum that we've gathered here. So I think lastly, of course, then it's important to thank all of you who've taken the time over the last 2 days to make this meeting work. The talks, I think, have been a significant improvement from last year. It's great to see all the posters up there, and we hope that next year there'll be even more, and the quality will continue to increase and improve. So thank you very much to everybody.
We work on a number of emerging pathogens and pathogens that are resident in Kenya and the region, including influenza. So our focus is really to help build capacity in the region, focusing in Kenya, both on laboratory capacity but also on epidemiology. So we're What we do is work with the ministry and other partners, including KEMRI, to understand burden of disease for all the pathogens that I've mentioned, including influenza. We work on a variety, dengue, rabies, and other pathogens, trying to understand the burden of disease with an ultimate goal to prevent, to identify intervention strategies, pharmaceutical like vaccinations or non-pharmaceutical interventions to help reduce the burden of disease. So the information we generate that's with the Ministry of Public Health Sanitation and with KEMRI can be used not only in Kenya but globally to help stem some of these infectious diseases.
Arboviruses cause diseases in humans and in animals, especially livestock, which human populations depend on. We have diseases like Rift Valley fever, which affects both human beings and livestock. We have a disease like yellow fever that causes very severe disease in human beings. We have a disease like dengue fever that also causes very severe disease and mortalities amongst human populations. So all these are viruses that are very important and do occur in form of epidemics. And when we have epidemics in our country, it causes a lot of stress and panic, and our people lose lives, and they incur a lot of losses in terms of deaths in livestock.
I work on the respiratory syncytial virus. This is an extremely important virus that affects infants and to a certain extent elderly adults. It causes devastating bouts of lower respiratory tract infection that often manifest in severe or very severe pneumonia. And it's absolutely critical that we find ways of limiting the ability of this virus to cause devastating illness. The basis of my PhD was to find out the relationship between the development of immunity against this paradoxical maintenance of the virus within the population.
The current African swine fever virus that is circulating in Tanzania has been described since the 1990s to be circulating in Mozambique, Zambia only. And then this virus was carried to Madagascar and then to Russia. But it's the same virus that has circulated to Malawi and then has been introduced via the southern border to Tanzania. And that means it's of very importance to the region, because if we don't work and control the disease in Tanzania, it could slip to Kenya. Our previous work, for example, in 2009, when we had an outbreak in Longido in northern Tanzania, we traced the outbreaks to be circulating at the same time with the similar virus, which is 100% genetically similar to another virus that was circulating in southern Kenya. So you can see that if there is leaks in the borders, the disease is going to circulate from Tanzania and probably to Kenya or other regions, other countries within the region.
Diseases have no borders, and what happens in Uganda can happen in Kenya, can happen in Tanzania. When we had the outbreak in Uganda, there was heightened alert in Kenya. So we advise our neighbors that they should be on high alert. They should improve on awareness to the health workers, because most often the health workers, when an infection occurs, people go to the health units. And if there is low level of suspicion, then many of the health workers will die. So we advise that the health workers may be put on the alert, may be trained, PPEs may be provided to these health workers, and let there be more training for lab personnel. Let the labs be ready to receive many samples and work hard hours. Let isolation wards be put in place, and let us collaborate.
We do surveillance activities for various number of viruses. We started way back in 2006 at the helm of the threats of influenza viruses, and we have since expanded to look at other pandemic threats, hantaviruses, arboviruses, filoviruses, a whole range of viruses that we look at.